Hi, my name is Mr. Chipman, and I teach AP Biology at Murray High School in Murray, Kentucky. And if you're looking to clear up common misconceptions to get AP Bio exam ready, maybe even enjoy a little biology along the way, you're in the right place. Before we get started, please don't forget to hit the subscribe button, turn on those notifications so that you can stay on top of every video in this series. We're going to be diving right into the topic here of mutations, dealing with common misconceptions with those mutations. Some of the concepts are a little tricky, and so we'll break it down together. Let's get started. So a couple mutations to kind of talk about the general concepts here. So first we're going to be looking at this idea of point mutations. Anytime you see the word point mutation, you're dealing with a single base substitution. Right, so what do you mean by that? We've taken, one has been taken out, another has been put in its place. There's no moving. Everything is just, it's like one for one, right? And so you can see here on these pictures, here you have TTC, instead you have TTT, right? Or whatever, you get the idea, right? And so there are three common point mutations that we'll look at. And we'll look at the two different types of missense mutations, even though I don't know that that's covered in the content as much. So three types of point mutations are, one of them is called a silent mutation. Why is it called a silent mutation? Still a mutation, technically a change in the DNA is a mutation. And so you have, instead of TTC, you have TTT, right, which leads to the messenger RNA being AAA instead of AAG, but it so happens that AAG and AAA both code for the same amino acid lysine. So since there's no change in the amino acid, there's no change in the shape of the protein, no change in the function. This is a silent mutation, meaning it's not saying anything, right? It's not affecting anything. A nonsense mutation, however, does create a change. Instead of TTC, you have ATC, which leads to the mRNA UAG, which is one of three stop codons, which causes an early stop codon, causes the polypeptide to break off prematurely, making a bad protein shape, which is going to disrupt function. So this creates this is a nonsense mutation, which creates a faulty protein. A missense mutation is when there's a single base that causes a change in the amino acid. And so you see the conservative versus non-conservative here. I'm just going to briefly hit on it. Conservative means that it's the same type of amino acid. It could have an effect on the shape of the protein, but it's going to be less. Whereas the non-conservative means that it's completely different, right? Instead of basic, you get polar. Instead of non-polar, you get acidic or whatever. All right, those types of amino acids, not so much important, but just hitting on the idea that there are different kinds of missense. But I think if you just understand missense being a different amino acid and that then affecting the shape of the protein, you're going to be fine. There's another type of mutation that we need to look out for, and it's called a frameshift mutation. And it happens from an insertion or a deletion, where a portion of the DNA is either been inserted into the hole or been taken out of the hole. And this causes everything to kind of shift over, right, one way or the other, which is going to cause the reading frame, which is in groups of threes, to be looked at differently. And so you can see here's the normal sequence here, and down here, by inserting a single T, you then kind of shift everything over to the right, and then which amino acids are going to be different after that? All of them. And so frameshift creates a significant change in the protein. All of that to say, it's very important that you get those things aligned, but there are some very important things that we need to deal with. One very common misconception concerning mutations is that they're all necessarily harmful, and we're going to be looking at that in a moment. But I think a connection here is to make sure that we understand that if there is a change in the genotype, then there will be a change in the phenotype, right? And so what could cause a change in the genotype? Well, a mutation could, right? Because the genotype represents the actual genetic makeup. A change in the genetic makeup, which is a mutation by definition, would cause then a change in the phenotype. Now, this phenotype could be negative or it could be positive. We're going to look in just a moment at how that is. Now, I think it's important also to note that that mutations don't necessarily mean disease, right? And a lot of times those two terms are sort of linked, or that disease doesn't always mean mutation. All diseases aren't linked to mutation. There are what are usually called genetic disorders, and a genetic disorder is sometimes linked to mutation, and it's usually going to be specific. It's going to say such and such base pair, this or this amino acid instead of that one, with like something like cystic fibrosis. Sickle cell anemia is another one that's commonly used in AP literature, where there's a single base pair that's changed, causes a single change in amino acid, right? But then some genetic disorders aren't necessarily caused by a mutation. There could be other things going on, as you see here, some epigenetic or some sort of environmental change. And so I think it's important to make those distinctions. When you hear the word mutation, don't automatically make other assumptions. Make sure that you're clearly reading the prompt, and so that you know what the kind of the follow through on that is. And so let's answer the question then concerning whether or not mutations can be beneficial. And so here's the very common example of the peppered moth. The peppered moth was a white moth that had sort of a black speckled on its phenotype, which is there on the left, which was great. If you were existing in a time where there were white trees that had little black splotches on them, you were perfectly camouflaged. And famously, these trees became covered in soot during the Industrial Revolution in England. And so the white moths stood out like a sore thumb and were being eaten by the surrounding birds that had a taste for peppered moths. And there was a mutation that caused the phenotype to be dark, as you see on the right. And those those moths were particularly suited for their new environment, right? So this would have been a positive mutation. It's a mutation in that there was a change in the DNA that caused a change in the phenotype, which we already looked at. But that particular change in phenotype was suited for its environment. Now, if the change in phenotype had been wingless or legless or something like that, obviously that's not going to be very well suited if you're a moth. But changing your wings to be black when all the trees are also black is particularly beneficial. And so whether or not a mutation is beneficial is going to be whether or not that change is then suited for the environment that it finds itself in. And I think that's a very important distinction. Make sure that you are reading the prompt carefully and that you're being able to draw those conclusions from it. Now, one other thing that needs to be mentioned is that DNA, or that cells have mechanisms for mutation repair. It's kind of built into the way that they work. And so during replication, there's several enzymes that do some checking, and if there is a problem found in the like A's, T's, C's, and G's, those particular sections can be cut out and can be gotten rid of. And there's another misconception that deals with the idea of coding sections versus non-coding sections of DNA, like the introns and exons and that sort of thing. The non-coding portions of the DNA aren't necessarily junk DNA, right? They could be used as regulatory sequences. They could be used as promoters, operators, all that sort of stuff that doesn't necessarily code for a protein, but is an important part of some sort of other function in the DNA. So be careful when you talk about non-coding portions of the DNA. Be careful the type of language that you use, that you don't call them junk or useless or something like that, but make sure that you are careful and precise with your language about those proteins. Hopefully this was helpful. If you found this helpful, please give me a like. If you have questions about this topic or any other topic, please give me a comment down below. If you'd like to see me address a particular misconception, also give me a comment down below. Thanks for watching. Really appreciate it.